Okay, well, I guess I'll just introduce myself here. So, I think most of you know who I am, Grant Hatch. So I've changed the titles a little bit, linking of cardiolipin remodeling to mitochondrial beta-oxidation. And I want to talk about some of our recent work on cardiolipin, its role in T-cell function. For those of you that don't know where I'm from Winnipeg here. We're the geographical center of North America. This is what we look like in wintertime. This is what it looks like right now. It's a cosmopolitan city, about 700,000 people. Nice place to live, a little cold in wintertime. I work in the Manitoba Institute of Child Health here. It's a new building that's been built in just the last few years. This is a wonderful facility where we have interactions between clinician scientists as well as basic research scientists. My office and lab are right here, and all of the basic science labs are on that fifth floor over here, and right over here are the clinical labs, so we have a great deal of interaction that can go on. This is the synopsis of what I'll talk about. I'll be very brief with my introduction to cardiolipin because we've beaten it a bit to death. I want to also talk about the role of this unique protein, stomatin-like 2 protein, in altering cardiolipin mitochondrial function in T cells, and also then go into the initial title of my talk, which is the role of the subunit of alpha functional protein in the cardiolipin remodeling. Fred Voss gave us a nice overview of this unique phospholipid cardiolipin, so I'm not going to go into that. Major membrane phospholipid, about 21-22% of the inner mitochondrial membrane mass, and synthesized exclusively on the mitochondrial inner membrane, only phospholipid to be done so. We know it's required for activation of enzymes of electron transport chain, supercomplex assembly. We know it plays an important role in protein and lipid important to the mitochondria. Some interesting studies with adriamycin have showed a cardiotoxicity related to this. So in addition to the anti-cancer drug adriamycin in intercalating DNA and preventing spread of cancer and development of cancer, it can also be toxic to mitochondria. And that's probably one of the reasons why you have a cardiac toxicity when you have adriamycin therapy. Of course, it's a regulator of apoptosis. It supports that caspase 8 activation platform. This is the mammalian cardiolipin biosynthesis de novo pathway here. Steps from phosphatidic acid down to phosphatidylglycerol that were described in the 60s by Eugene Kennedy. Many people in this room have been involved with cloning and sequencing of these genes. The final step of the pathway is conversion of phosphatidylglycerol to cardiolipin catalyzed by cardiolipin synthase, and that was characterized by Carl Hostetler when he worked in the laboratory of the great Lawrence van Dienen. This is an excerpt from Michael's paper on cardiolipin molecular species. Of course, there's an enrichment, particularly in the heart. About 80% is tetralinoleal cardiolipin. And because the cardiolipin biosynthetic enzymes are rather nonspecific in terms of putting on the appropriate fatty acids onto the cardiolipin molecule, we need these remodeling pathways. Roger Shih is here, and he's the pioneer of the studies on ALCAT1, which is involved in the remodeling of monolysis of cardiolipin in the ER to form cardiolipin. Michael has, of course, characterized the functional role of tafazin as a PC and PE donating linoleic acid. We're going to hear some interesting work from Toon de Kroon later about phosphatidylserine role in yeast. We did some work on monolysis of cardiolipin acyltransferase 1. It basically has the same activity as the monolysis of cardiolipin acyltransferase, or ALCAT1, in the ER, except it's in the mitochondria. And then recently, the Hemmings group in Switzerland has demonstrated that this protein THEM5 could be involved in remodeling of the cardiolipin. So I'll turn now to that first part, the role of stomatin-like protein in altering cardiolipin mitochondrial function in T cells. Stomatin-like protein, which I'm going to refer to as SLP2 from here on in, is a member of the stomatin prohibitin flotilin HFLC/K superfamily of proteins. They're a highly conserved family of proteins that mediate interaction with cell membrane, upregulated in a few cancers, modulates mitochondrial membrane permeability transition, and also ATP production. If you take SLP2 cells, if you take peripheral blood mononuclear cells and hit them with 4-ball ester and inomycin, which is in essence blasting the cells, you can dramatically upregulate the expression of SLP2 protein. Very interestingly, if you look at the immunological synapse, this is a Jurkat T cell activation, for example. If we have green fluorescent protein expressing SLP2 Jurkat T cell here, we can see that there's some localization of the SLP2 in the periphery here. CD3 is a marker for the T cell receptor on the plasma membrane. put them together, you can sort of see that the SLP2 and the CD3 don't come together. But upon activation with staphylococcal enterotoxin E, you activate the T cell, you see something rather remarkable. You see all the SLP2 lining up right at the immunological synapse, along with the CD3, which is the marker for the T cell receptor signalosome. And interestingly, if you take a look at more gross mitochondrial pictures, you'll find that the mitochondria kind of line up right here at the immunological synapse. So it's quite unique. And this was shown by Quim Madrinas' laboratory in a recent paper published in PLOS One. Quim's also shown that SLP2 expression increases effector responses, whereas downregulation of it correlates with loss of T cell receptor signaling and its activation. So we were able to obtain some SLP2 green fluorescent proteins, SLP2 Jurkat cells that stably express SLP2 green fluorescent protein. And you can see that it looks like this. When you add mitotracker red to that cells, you can see that they're almost on top of each other. We do the merge and certainly see they're exactly in the mitochondria here. And this is just a three-dimensional picture. If one looks at two cell lines that were generated, a SLP2 low-expressing cell line under doxycycline promoter, which essentially has the same amount of SLP2 protein as the parental cell line, compare that to a SLP2 high-expressor cell line. When you add doxycycline, you get an upregulation of SLP2 green fluorescent protein. You have a lot more here, about a 3.5-fold increase in SLP2 protein. It's exclusively mitochondrial, and this is just a marker of complex 3. Now, expression of SLP2 in T cells increases metabolic active mitochondria. This is the SLP2 low-expressing cells treated with doxycycline, mitotracker red staining there. The high-expressing cells, you get a much more wider band of the mitotracker red staining. Quantitating this, the mitotracker red, you can see that it's elevated quite dramatically in the SLP2 high versus the low. And if you actually count the number of mitochondria per cell slice, you can also see it's upregulated in SLP2 high versus low cells. It seems to increase cardiolipin and mitochondrial biogenesis. So when you SLP2 high expressing cells, looking at the actual mass of cardiolipin increased about 37%. Non-acridal and orange staining increases a bit. Acetate incorporation of cardiolipin increased. Linoleic acid incorporation of cardiolipin is increased when you incubate those cells with linoleic acid. mRNA expression of cardiolipin biosynthetic genes, for example, phosphoglycerol phosphate synthase, cardiolipin synthase are elevated, and the master regulator, as Mingdong had mentioned before in his previous talk, PGC1 alpha, is dramatically upregulated by SLP2 expression in the T cells, and mitochondrial DNA as well increased 70%. Another interesting thing is that SLP2 actually binds to cardiolipin. So if we do one of these pull-down assays, we take vesicles of cardiolipin and you incubate them with human recombinant SLP2. And when you treat them with sodium carbonate, centrifuge them down. If it binds actually to the lipid, it'll end up in the particulate fraction. If it doesn't, it'll stay in the soluble fraction. So cardiolipin, you can see, nicely pulls down SLP2. The other phospholipids, PC, PE, PI, do not do that. Phosphoglycerol a little bit because it's sort of essentially half of a cardiolipin molecule. And interesting here, just looking at this slide here, the different ratios as you increase the amount of cardiolipin to PC, PE ratio, you see a proportionate increase in the amount of SLP2 that you're able to precipitate out into the particulate fraction. SLP2 also interacts with prohibitins. So, for example, if we take white blood cell lysates, and we do a SLP2 immunoprecipitation, and then do Western blots for prohibitin 1 and prohibitin 2, we can see that it interacts directly with these two prohibitins, which are in the mitochondria. Another experiment which you can do is you can take the whole cell lysates and you incubate with increasing amounts of doxycycline to progressively increase the amount of SLP2 green fluorescent protein, and then do a SLP2 immunoprecipitation. And what you'll find is that you have a loss of prohibitin 1, prohibitin 2 from the whole cell lysate, and an increase in the amount that's associated with the SLP2 as you increase the amount of protein, which makes a lot of sense if it interacts. SLP2 expression in T cells will stimulate mitochondrial activity and interleukin-2 secretion in response to T cell stimulation. This is citrate synthase activity, NADH dehydrogenase activity, succinate dehydrogenase activity. ATP production in both mitochondrial as well as the whole cell is increased by the induction of the SLP2. Oxygen utilization is particularly evident when we have low amounts of glucose in the medium here. You can see a dramatic elevation in oxygen utilized by these cells. And importantly here, when we look at interleukin-2 production, which is the activation of the T cells, in the SLP2 high-inducing cells incubated with staphylococcal enterotoxin E, their ability to respond to stimuli, i.e. generate interleukin-2, seems to be elevated. Well, what about the corollary, knocking it out? First of all, SLP2 knockouts are embryonically lethal at the implantation stage. So what we did was we did a Crelox approach to reduce the SLP2, and basically we're able to delete SLP2. It's under the CD4 promoter. And this is just a Western blot to show that in naive T cells or blasted T cells, you don't see any SLP2. The number of white cells seem to be fairly normal, thymus, spleen, and lymph nodes. There appears to be, if you look at the lymph nodes, which kind of looks like a tiny decrease, but it's not statistically significant, there seems to be a decrease in the CD4 T cells, a little bit of CD3, but certainly more in the CD4, although this is not statistically significant. But there does appear to be a reduction, slight reduction in the CD4 cells. The T cells that are deficient in SLP2, they exhibit reduced mitochondrial complex protein activities, insoluble prohibinin-1 and cardiolipin. Basically, just looking over here, you can see in the knockouts, there's a reduction in complex 1, complex 2. There's another complex subunit here, complex 4, slightly reduced. These are other complex 1 proteins. There's a whole plethora of these, but we didn't analyze all of them. If you take the mitochondria and you do a triton-insoluble extraction and look for prohibitins in the knockout insoluble fraction, you can see that that's reduced. This is indicative of the electron transport chain complexes that one might see. Complex 1 activity is reduced, succinate cytochrome C oxidoreductase activity reduced, and cardiolipin in that insoluble fraction is reduced as well. Finally, we looked at T-cell specific knockout, what it does in terms of responsiveness. We've seen here that if you take the white cells out of these knockout animals and hit them with staphylococcal enterotoxin E, that in the knockouts, their ability to produce interleukin-2 is reduced. If you take then these animals and immunize them with the ovalbumin, wait 10 days, then take those white cells out again and hit them with increasing concentrations of ovalbumin to stimulate a response, you see that the response is blunted in the knockouts compared to the wild-type animals. And finally, when we take our knockout animals here, either sham operate them or operate them and put on an ectopic heart and look for allograft rejection, we can see that in the wild-type, rejection occurs at this time of between 7.5 and 9.5 days. In the knockout, the ability to reject the heart is reduced by about a day, or delayed by about a day, day and a half. So to summarize, SLP2 expression in T cells stimulates cardiolipin and mitochondrial biogenesis activity and T cell ability to respond to stimuli. And then SLP2 knockout in T cells reduces cardiolipin in prohibitin complex fractions, mitochondrial metabolism, and T cell ability to respond to stimuli, which links cardiolipin to T cell function. And we're hypothesizing that SLP2 functions to recruit prohibitins to cardiolipin-rich microdomains in which electron transport complexes can be optimally assembled for generation of ATP. So the last part of the talk is the role of alpha subunit of human alpha trifunctional protein in cardiolipin remodeling. A number of years ago, we'd shown that we purified and cloned sequenced this monolysis cardiolipin transferase enzyme and shown that when we express it in Barth syndrome lymphoblasts, we got a dramatic upregulation in the cardiolipin mass. At that time, we didn't look at the tetralinolale species, but we also see an increase in complex 2 and 3 activity in the cells that express the monolysis cardiolipin transferase. So when we look at the sequence of human monolysocardiolipin transferase, it's basically a shortened or a truncated version, a 59 kilodalton version of the alpha subunit of the human alpha trifunctional protein, the 74 kilodalton protein. This is not a very good picture, so we'll just blow it up a little bit here. Trifunctional protein, as you know, has these 3 enzyme activities in beta-oxidation, heterocomplex 4-alpha, 4-beta. Looking at the first part of the NP00173, which is the accession number for alpha subunit of trifunctional protein, the transit peptide region. If you go further down here, you can see the same thing in the pre-monolysis cardiolipin, and the rest of these molecules are identical. So we thought, since we'd shown that this monolysis cardiolipin transferase has enzyme activity for remodeling of cardiolipin, what about alpha subunit of trifunctional protein? So we generated human recombinant alpha subunit trifunctional protein with a histidine tag, shows up nicely here at 75 kilodalton. A generous gift from Jacques Couture. His antibody, anti-trifunctional protein antibody, binds quite nicely, just proving that we have activity, or excuse me, we have the protein. We then incubated this recombinant protein with either palmitoyl coenzyme A, linoleal coenzyme A, or oleoyl coenzyme A or linoleal coenzyme A radio-labeled, and look for the formation of cardiolipin, and we can see that there's formation of cardiolipin. Looks like it's a little better for 18.2. Kms and Vmaxs are down here, about 18 for the linoleal coenzyme A as substrate. This is just a mixed monolysocardiolipin, a bovine heart cardiolipin mixture. Kms a little bit higher for the oleoyl and palmitoyl, and the Vmax about 6,600 picomole here, 716, 795. And then if we take the HeLa cells and we either incubate them with radioactive palmitate, oleate, or linoleate, transfect those cells with the alpha subunit of trifunctional protein, we can see that we can stimulate the actual formation of radio-labeled cardiolipin compared to the mock transfected cells. You'll have to trust me here. This is about a 30% increase with the palmitate and the oleate, and linoleate is here. It seems like to be a little bit more specific for the oleate, the linoleic acid remodeling process. We think and we believe that this monolysocardiolipin acyltransferase is likely a splice variant of the alpha subunit of trifunctional protein, because what we've done here is that we've generated RNAi, inhibitory RNA sequences to this part of the molecule here of trifunctional and this part of the molecule here of trifunctional, and looked for the expression of monolysocardiolipin acyltransferase in cells that have been transfected with the RNAi. So in the HeLa cells that we transfect the on sequences here, we can see that there's actually a reduction in alpha subunit of trifunctional protein. Confirm that by real-time PCR. But when we look at monolysocardiolipin acyltransferase expression, no change there, and there's no change when we look at real-time PCR. So we're thinking along the lines that it could be a splice variant. And there's some precedent for that, actually. Finally, we're looking at the expression of monolysocardiolipin acyltransferase or alpha subunit of trifunctional protein and how it affects or modulates tetralineal cardiolipin formation. Here, this is control lymphoblasts. These are Barth syndrome lymphoblasts down here. Alpha subunit or monolyso will increase mRNA expression when you transfect it, not surprising. Same thing here in the Barth cells. We're able to express both of these. We've seen this before, of course, for monolyso. And here, we look at the tetralineal cardiolipin species, either in the normal cells or the Barth syndrome lymphoblasts. They're elevated by both the 59 kilodalton monolysocardiolipin acyltransferase or the 74 kilodalton trifunctional protein. It's much more higher here because you have a lower amount of cardiolipin, tetralineal cardiolipin to start with in the Barth cells. The enzyme seems to be very specific, doesn't seem to use any other lysophospholipid substrate that we've identified other than monolysocardiolipin to acylate to cardiolipin. And finally, we just did this experiment for a lot of fun. We wanted to check for the ability of knocking down TAZ in normal cells, normal lymphoblasts, to see if monolysocardiolipin acyltransferase could compensate for the loss of tafazin. And in this cell, what we've done, these are again normal human lymphoblasts. We've looked at monolysocardiolipin acyltransferase by adding inhibitory RNA for TAZ. We basically see no effect on monolysocardiolipin acyltransferase activity. But we can, when we add a monolysocardiolipin acyltransferase 1 plasmid transfected, we can increase activity as expected here. But this is quite unique here. In the normal lymphoblasts, when we hit these cells with TAZ RNAi, incubate the cells with radioactive linoleic acid, and look for the percentage of radioactivity in cardiolipin, or in cardiolipin with respect to the cells, we can see that it's reduced in the TAZ knockdown. And then when we add monolysocardiolipin acyltransferase plasmid back to these cells, we restore that back to normal. So to summarize, we believe that alpha subunit of the trifunctional protein exhibits monolysocardiolipin acyltransferase activity, and that expression of it stimulates cardiolipin remodeling with oleate, increases L4 cardiolipin levels in both normal as well as Barth syndrome lymphoblasts, now linking an enzyme of beta-oxidation to cardiolipin remodeling. And also, we believe that monolysocardiolipin acyltransferase is not increased by TAZ knockdown, indicating that TAZ and the monolysocardiolipin acyltransferase probably don't complement each other in cardiolipin remodeling, but at least we believe that the enzyme its expression can compensate for loss of TAZ. So currently what we're doing right now is we've managed to get a hold of the tafazin knockdown mouse, and we're growing it up, and Laura Cole in the laboratory is generating a lentivirus for both monolysocardial acetyltransferase 1 and alpha trifunctional protein, and we're seeing if expression of these in these animals will be able to attenuate the development of cardiac defects, and we're also going to look at other organs as well, including the liver. And finally, we want to look at whether there's a SLP2 protein plays any role in mitochondrial dysfunction in Barth syndrome. We're going to screen for that, and we want to see if SLP2 expression in Barth syndrome can actually increase mitochondrial biogenesis, potentially protecting the cells. Although, as we've heard today, upregulating bad mitochondria might not necessarily be a good thing, but at least we know that we can upregulate PGC alpha 1 with SLP2 expression. So just to acknowledge the people that are involved with the work: Bill Taylor, Fred Zhu in the laboratory, Edgar Mejia, new graduate student, Laura Cole, a bright postdoc who just came over from Dennis Vance's laboratory to work with us on the animal work now. Derek Christie, a bright PhD student who's now graduated from Quim Madrinas' laboratory, formerly at Robarts. Now he's at McGill, chair of immunology there. Jen Spragna and Rob Murphy were involved in measuring the tetralineal cardiolipin species for us. Korshaja for his generous gift of the alpha trifunctional protein antibody. Thank you. And the funding agencies, and especially this little guy here. Okay.
Thank you, Grant. Questions for Grant?
Yeah. Yeah.
Do you think that the SLP2 effect is the absence of SLP2 is affecting the generation and/or longevity of memory T cells?
One of the things that we have observed is that the ability of the T cells to differentiate into T helpers like T1 to T17 lineage is not affected. In addition, if you look at production of things like interferons, they're not affected as well. It seems like there's some specificity for interleukin-2 production being affected. So that's all really the information that we have at this point in time. We haven't gone back to deal with that a little bit more exclusively. Yeah.
Grant, beautiful work. I'm just always wondering, so far we have only one cardiolipin synthase being identified.
Right.
Could the SLP2 be one of the cardiolipin synthase isoform? Have you ever done, by knockout cardiolipin synthase, by overexpress this one, and it can complement the defect? That's question number one. Number two is MLCL AT always amazes me. Did you ever analyze the activity towards dialysyl cardiolipin, whether it's still exclusively recognized MLCL?
Yeah, I'll answer that first one. We did in the JBC paper, we had looked at dialysyl cardiolipin, and I think we just made a statement like data not shown that there wasn't very much activity at all for that. So we weren't able to see that. And your first question again was
SLP2, whether it has cardiolipin synthase activity.
I mean, if you look at the structure of SLP2 and look at its sequence homology, it doesn't seem to be homologous with what we know of the structure of cardiolipin syntheses. Now that doesn't mean anything. It could potentially have that catalytic activity. We've never looked at that, but that would be something that might be worth looking at.
Because so far, all the phospholipid remodeling enzyme has multiple members. Like GPAT has 6.
Right.
LP, LPC, LPS, LPI, all the remodeling enzyme has multiple members. Yeah. Cardiolipin synthase, only one. It's such an important enzyme.
So my question is SLP2 bind to cardiolipin. So is there any consensus cardiolipin binding domain or SLP2 or any other proteins? Yeah,
there are lipid binding domains, consensus sequences for lipid binding domains. But the unique thing, I guess the way the molecule is structured, only cardiolipin seems to fit into that cavity or will interact with that, which is quite surprising because so there must be, in addition to the acyl chain specificity, there has to be something to do with electrostatic charge or electrical charge. Because if you notice phosphatidylglycerol, which is basically half of a cardiolipin molecule, seem to be able to bind to a certain extent into that, whatever site that is. Yeah. Michael?
When you assay the alpha subunit for acetyltransferase activity, did you look at this turnover number problem that we discussed like 2 years ago?
No, we didn't. Actually, at this point in time, no.
This was a critical point, right?
Critical point,
that the actual turnover number that you could calculate from the data of your purified enzyme was extremely low.
Right, yeah.
I mean, which could have multiple reasons, of course. It could have something to do with the assay conditions or with the fact that the activity of that you're measuring is not really a bona fide activity of the enzyme, but some sort of, well, let's call it side activity or secondary activity that you perhaps also could generate with other proteins. But in any case, that is sort of a problem, I think, that needs to be, that perhaps should be followed up on.
Okay, I think we do probably need to do that experiment, certainly. I mean, when we look at the kinetics in terms of the...
It should be obvious from the data, I mean, because you know how much you added, right? So you could back calculate this from your data.
Yeah, but like you say, the calculated activity is actually quite low. And that probably explains why it can't compensate for loss of TAS. If anything, it could have a maximum of 20% cardiolipin remodeling under normal circumstances. But again, TAS probably accounts for the majority, probably greater than 80% of the actual remodeling activity in vivo. Yeah.
Grant, there are still a few problems remaining with this notion that the alpha subunit might do the job as you think it does. First of all, the alpha subunit, we have patients, we discussed this before, you have patients which are fully deficient in the alpha and beta subunit of this MTP complex, and they have normal cardiolipin levels. That's one. Right. The other thing is that the alpha subunit, if you express it, it's completely unstable in the absence of the beta subunit. So I wonder, in your case, how that would work out for your splice form. Arnie's looking at me. So
yeah. Well, the splice again. I mean, in terms of stability, I mean, after production of a human recombinant enzyme, it seems to be relatively stable when we put it in a membrane environment. So.
Yeah, but it's not really alpha subunit. It's only 2/3.
It's only 2/3. Yeah, it's 59
kilodalton.
So
it doesn't have to be in the same place, and we may not even be detecting it in some of the patients.
Right.
And it doesn't have to have the same promoter either. So.
Right.
I mean, we have some alpha subunit knockout mice that would be very interesting to look at.
Okay. Be interested in looking at that, certainly. Okay, any other questions? I guess if not, how long do we have for a break?
We have 15 minutes.
Okay, 15 minutes, so we'll be back here in